I have introduced Steve, and let Steve introduce himself.
Or not, because... yeah. Okay, so they're starting you guys off easy by having me with this, because I'm going to tell you stuff you already know. So no depth, right? Okay, so it's this, not kidding. So, when we were working on this at the Garbalata lab a while ago, we got a lot of different ideas, and some of these things were, you know, pretty proven, and they just needed to be tested out a little bit, something like agrophos. And, you know, some other things you've already heard about, foliar host removal, things along those lines. And I'm not going to talk about those either. But we also got some less conventional ideas. So wind chimes was one of them, and we didn't test that. But I am going to talk to you about at least one more. So this idea of bark scribing, can you see what that is on the screen? It looks kind of weak from here, but that's all right. So the idea here was, in the early 2000s, we set about trying to figure, before we really understood the role Bay laurel had, we set about trying to figure out what oaks were infected and what oaks weren't. So we were cutting into the bark about, you know, just deep enough to be able to take some tissue samples, isolating them and seeing if we could grow it out on Norma, right? And when we visited those same trees that we had sampled from about a year later, we realized that a lot of these trees were forming callus tissues and really had stopped. It looked like we had stopped the infection when we had put it up there. And so we got this hypothesis, right? And I wasn't the only person to think this. Hey, what about, is it possible? Phytophthora likes things, what? Cool, dark, moist, and when you cut it open, it makes it bright, dry, and warm. So maybe it's just enough to push it to the point where the oak could fight it off and everything. It makes perfect sense, right? And this is what it looks like when you go back and visit it the next year. You know, no problem. So, you know, you look it up, you're thinking, well, maybe that's kind of far-fetched hypothesis. But then you go and you look up IPM for citrus, and you look up Phytophthora infections on citrus trees, and you see IPM has it listed as a treatment for other Phytophthora species. You just go in, you do some bark scribing, and they've been using this for 100 years, right? So you're saying, well, now it's kind of of course, you can't find the literature on it. Oops. But, you know, it's kind of a no-brainer that, so if you're not familiar with what bark scribing is, you know, we cut about halfway through, as you see on the right-hand side, beneath the bleeding area, and that's the idea. You're not trying to cut it out. You're just trying to change the environment that the pathogen's working in to be less favorable to it. So, my predecessor went out and he decided he wanted to test this. You know, so he treated about 200 trees on private properties, and the problem is, how do you figure out what size the lesion is to compare? As soon as you cut into the tree, it's now a treatment and not a control. So there's not a really easy way to control for this. You could, of course, inoculate trees, but you'd have to have a healthy tree for that. So, hey, can we take this deadly pathogen and put it into your healthy tree and then see how it does? A lot of people aren't signing up for that one. So, when he ran these, he started looking at it and he said, well, it looks like we get pretty decent control. It's sort of a cancer analogy. If we can catch it early enough and we can change the environment early enough, then it seems to work. And if it's too advanced, the lesions are too big, obviously you're not going to be able to change the stuff or the entire lesion, and there's a chance something may survive. Okay, so this is one of the trees that he did, and this is where, so you can see he cut away, and then if you walk around the tree just a little bit, you'll find this little bleeding spot where they missed one spot. So this is sort of that same hypothesis we were talking about, the cancer hypothesis. It was just too advanced. We couldn't stop it. Oh, sorry. Right? So when the industry finds out that, well, and people find out that, gosh, they're doing all this bark scribing work on a couple hundred trees in the Marin area, they all want it too because it's the new experimental thing. It's very sexy. So, and so tree services started offering this too. And this is not a criticism. I mean, people wanted it and they provided it. That's okay. Although some of them called it an experimental treatment, and it really wasn't because nobody's going back and checking it out. And agrophos didn't always work, so people were starting to, you know, it had been touted as this big thing, and people had been spraying trees for several years, and they still died, and they weren't happy about that. And some of them did really nice artwork. I mean, this is fine work that we're doing. Okay, anyway, so we wanted to test that. So we got about 60 trees, 3 sites, and we infected branches on mature trees because nobody would let us do the work directly on the trunk. Good, I'm all right. And so we put in positive controls, negative controls, and treatments, and a couple backups. Pretty straightforward. Then we checked the progress of the infection, see how they were growing with the backups, and we planned it to take 1 year, but of course things don't work out the way you want them to, and it took 2. We harvested our positive controls a year after that, and our results were doing nothing works great. So lots of positive controls failed to thrive, and lots of treatments failed to thrive. And there was no statistical difference between them. Except when it doesn't, of course, and that was when we lost a whole bunch of trees because they just croaked. So what it ended up being is we lost more than a quarter of our positive controls or our treatments. We lost more than a quarter of them. The tree killed it just fine. We lost another roughly quarter of it with the beetles come in and take it out, and they just up and die. And this is consistent with a bunch of other talks you've heard here today. Basically, when you do this bark scribing, it seems that we're taking credit for the trees defending themselves. When you cut into the tree and you find a small lesion, you didn't catch it when it was small. The tree had already stopped it. You are taking credit for, look at it. And when you find a huge lesion, it's not because it was, we got here too late, we could have saved it 2 months. No, the tree had no way of stopping it. So again, sorry, but if you'd called us before, we could have done it. Ultimately, there is no silver bullet, and I'm just here to remind everybody that even when the proven treatments only work marginally well, and I'm just reminding us that, as scientists, sometimes we get really attached to our ideas and don't believe everything we think. And as a follow-up for my time up, UC did remove their bark scribing from their recommendations for citrus. You can find it only in one place, and they're in the process of revising that now, Dan.
That's me. Here we go. Hi, I'm Lauralea. I'm very fortunate to be the lead trainer and handler for the Phytophthora project at the Garbolato lab. We are researching the possibility of detecting Phytophthora through odor. So this is Bolt, the lightning round. That's her name. Bolt. That's my name. Okay. So current methods, you all know this, I don't need to repeat it. The problems are large-scale detection on a commercial level, very difficult and expensive and time-consuming, so we're trying to find a way to sort of streamline this process. I have a heavy background in dogs, and I can tell you right now, they can help you quite a bit. So we're going to experiment with this concept of using scent. Dogs have been worked in ecology for a really long time, about 100 years. People don't realize how long dogs have been involved in wildlife and forensics. We've gone from invertebrates, birds, botanicals, obviously it's why I'm here. The cool one too is fish. We can actually detect fish in the water, so it's amazing what they can do. So it's a well-established field. This is not a shot in the dark kind of fly-by-night situation. So the capabilities of scent dogs for Phytophthora detection and management. They're economical compared to current testing methods. They're fast. Results are actually achieved in seconds or minutes. Lifetime detections instead of hours where you might need for certain testing or sampling or baiting, which could take a while. They're practical for large-scale production. They detect very quickly at high speed. It's efficient. Sample testing can be done on-site with minimal space. You can set up a quarantine area, run your samples through if we're at a production facility like a nursery, large-scale nursery. We can actually gather samples and run the dog across those samples in minutes and get accurate detections that way. They're precise. A well-trained dog can distinguish target odors from peripheral and contradictory odors with a very high accuracy rate that's done through training. And their sensitivity presence is very high. They can detect asymptomatic trees. We see this, I know there's a mention of cancer, totally ties in. Dogs have been proven to be able to find cancerous tumors in the human body well before they are clinically detectable, especially with things like colon cancer and lung cancer. Pretty amazing stuff. So our first method was phase 1, and our question was, can the dog even detect Phytophthora? And can the dog detect it across a range of media? So we chose 4 species for this, ramorum, cactorum, cinnamomum, and numerosa. The media we tested were mycelium and soil tea, and then we did 2 different types of soils with the mycelium. They were all grown in pea broth and incubated and prepared accordingly. The scent dog was trained to recognize target odor using classical conditioning. I actually use a clicker. Lots of food and toys. Show you a really quick video of her searching. There's containers on the floor. They're controls, so we have soil tea present. Only 1 jar contains the mycelium. This is a practice round for me. I don't know where it is. Tina's in the room with me, who's amazing, you don't know her. So she's going to let me know when I'm correct. So this is kind of a practice for our trial. So we run them all double blind. In this situation, it's single blind. So I'll let you see this. The target is actually right here. So this is the lab. That's Bolt. She's adorable. I'm also biased. She is a rescue dog, which is super cool. So she's clearing controls. She says, hey, wait. Bam. This is probably 4 days of training. Okay, she got it real quick. Okay, so we ran a bunch of trials. We did these double blind. We also did single blind because we like to do that. I feel like I'm leaning over. Is there a way to? We do our tests in 10 trials. It's 10 runs. So a run is we have 8 to 9 controls, whether it's the soil or the tea, whatever we're using for our substrate, and then we have 1 target available. Again, I don't know where it is coming in with the dog. It's confirmed verbally from Tina, who's outside the room. So we conducted it across the species, across the 4 different types of substrates, and we got a 100% detection rate, which were true positives on all. It was pretty awesome. In a very short amount of time. These tests took about 10 minutes to run. So the dog went through less than that. And that just, it takes time to move the jars around. that's what takes the most time. I'm waiting, I'm like, okay, let's go. You saw how fast the detection is. It's super quick. Phase 2. So our next question was, okay, we've proven that the dog can do that. Can she discriminate Phytophthora species across co-occurring and distracting scents? In detection, we call this proofing. It's really just clearing that picture for the dog that we want to target this specific odor. And so we throw a lot of things in the mix to sort of confuse the dog, and it teaches the dog to kind of tease out all those distracting odors and really find that target. They're really good at this. They are very good at being specific. If you own a dog, you know how hard it is to get them to sit anywhere but at home? Yeah, they don't generalize very well. So they're really good at being species specific. In fact, it takes a lot of training to get them to generalize. So we started with rhododendron plants. They were inoculated, the leaves with ramorum and numerosa, the roots with cinnamomum and cactorum, and then we also detached the leaves and did plugs to infect them separately and then incubated those. We proofed the dog. We also did a bunch of runs where I threw a bunch of just random stuff at her. I had toys and food and treats and controls, our gloves, the foil, anything that we were using that we were handling the samples with, we presented to her as well so she could work through all of those, and it was pretty fun. We tried to throw her off with Cheetos and beef jerky, and she wasn't having it, so it was pretty fun. Again, we used classical conditioning for this. I have a quick video, which one of my favorite terms as a trainer to use is odor obedience, and that's when the dog, despite whatever's happening in the world at the particular moment, when their target odor that's been conditioned is presented to them, they will respond to it. It becomes an unthinking response. The dog just will sort of, it's kind of like that cartoon, the Bugs Bunny cartoon with the carrot. Remember how it used to do with the scent thing? It would like go down the burrow and find Bugs Bunny and grab him by the nose and just pull him out. It's the same thing. And it's just a fun video, so I thought I'd show it. We weren't even working just yet, but the target was down, and she's like, oh, hey guys, cookie. And she's right. And I was like, free? Yes, okay. She took her ball over there. I thought it was really cute. Really cute, right? Don't ask what samples those were, because I do not remember. Okay, so our methods. Initially, we left the leaves on the plant, but we were concerned about cross-contamination, so we ended up using things like the jars to keep her from touching them. We also took root samples and put those in the jars as well. Again, ran the trials. You can see across the map, 100% detection rate on even those. I'm almost out of time. Our next phase, real quick, we're going to go further with this and do discrimination against Pythium. So we're sure that we're not having any confusion there. And we're also adding Tentaculata to the mix. Future goals, you can read that. We just want to really get this concept really hammered in. We want to really solidify our sampling methods and standardize the protocol. I'm really big on that. I think it's important that when you're using a method that's a little off the cuff, that you're doing it clean and that you're setting a standard for the future. So thank you very much. Thanks for having me.
Hi, I'm Norma Klein. I'm the OSU Extension Forester for Coos and Curry counties, and I'm presenting the initial findings of our project using citizen science and outreach education to reduce the risk of ramorum spread in Oregon's forests. And my project partners are Sarah and Jared, and this project was funded by a USDA APHIS grant. So we implemented a coordinated outreach and citizen science project to teach our participants the importance of early detection to slow the spread of sun milk death in Oregon. And we used three main outreach approaches, the first being workshops to promote the general awareness of sun milk death, and the second is our citizen science project, where we're focusing on early detection strategies at the leading edge of the disease. And the third is a project where we have citizen scientists searching for disease-resistant tan oak. And so our project area is in and around the quarantine area in Curry County, Oregon. So far, we have held 3 workshops and a hike to teach participants about sun oak death and to recruit citizen scientists to our project. Our workshops are a combination of talks and handheld activities. We then implemented our citizen science project, focusing on early detection strategies at the leading edge of the disease. And so we worked with one group of citizen scientists setting up bucket baits under tan oak in the rainy season, and we worked with another group of citizen scientists setting up string baits in the drier months. And the bait stations are located on the properties of the citizen scientists, and many of them are small woodland owners. So for the bucket baiting, we had 5 landowner sites. We monitored 2 tan oaks per site and 2 bucket baits per tan oaks. So we had 4 buckets per site. And the citizen scientists took samples every 2 weeks for a 3-month period. The stream baiting, we had 4 landowner sites, 1 bait station per stream, and once again, that was a 3-month time frame and more in the dry months. For both projects, we supplied clean bait leaves and supplies, and the citizen scientists took sampled the bait leaves and mailed them to the OSU Levoldis lab for further analysis. We also investigated rapid tests as a possible screening tool. So the volunteers set up additional bait stations at each sample site for on-site testing using the rapid test, those little kits. Our additional project was a project where we are training participants in the generally infested area to identify and report healthy tan oak. We worked with the Tree Snap app developers at the University of Kentucky and Tennessee to include our tan oak information in the Tree Snap app so that our citizen scientists could use the app to geolocate possible disease-resistant tan oak. So a few observations regarding working with volunteers. We found it difficult to recruit high numbers of volunteers in rural areas. Instead, it's probably more efficient to focus recruitment in strategic locations. And in our project, that was at the leading edge of the disease. We assisted volunteers with setup, and we provided them with detailed instructional booklets, and we felt that that really helped the progress go smoothly. In terms of project outcomes, our first season of bucket bathing, we did not have any detections of Phytophthora ramorum, but that's not surprising since the buckets were located in advance of the disease. And our stream bathing project is ongoing. But what we did find is that the citizen scientists, they followed through with a 3-month sampling project, so that's quite a commitment. And they followed sampling protocol. And in terms of the rapid tests, of course these tests are only specific to the genus, but it was a really great tool to engage our citizen scientists because they could use the test as something hands-on that they could use. But kind of an unexpected outcome was that for the string baits, where you're more likely to find random Phytophthora, it created a great deal of anxiety to our I had to really talk them down. Also, our folks are interested in future projects, so we didn't wear them out. I don't think this was a one-time deal. I think they're going to continue. And of course, citizen scientists are knowledge multipliers in the community. We had a number of our folks who recruited neighbors and friends to be part of our project, and they've been talking to lots of folks about it. And that's all I have. Thank you.
All right, I'm Devon Gaydos. I'm currently a PhD candidate at North Carolina State University, and I'm going to be talking about my work developing interactive disease simulations for Oregon. So part of what my lab specializes in is the development of these simulations which show how a pest or pathogen is likely to spread across the landscape. We can do a lot with these from a research perspective, but one of the most interesting things we can do is to manipulate the underlying host maps to simulate management. With that, we can test out a whole range of scenarios and see how that may affect long-term disease spread. And we think that that could be really useful for especially a place like Oregon that's doing a lot of active management. However, these models aren't necessarily accessible to the stakeholders who could benefit from them the most, especially the agencies who are doing this kind of management. And honestly, it's understandable because these typically have a lot of barriers to their use. They require a lot of coding, they require a lot of geospatial data, and there can be a pretty steep learning curve there. So what we're seeing is that there's this disconnect between the research and the on-the-ground decision-making, which is not uncommon for these kinds of computational models. One idea to overcome this problem is by developing more user-friendly interfaces. And importantly, there's this idea that we should be developing these interfaces in collaboration with the stakeholders who would ultimately be using the tool, which is this idea of participatory modeling. So as researchers, my understanding of the system is limited, especially because I live in North Carolina, not in Raleigh. So by working with stakeholders who have a different perspective, we can get a more holistic understanding of the system. And for the stakeholders, if they're engaged in how these tools are developed, then they're more likely to ultimately meet their needs. Honestly, all of this seems pretty straightforward, but this is still a fairly new concept in research, especially in the case of forest pests and pathogens. If you were here at the last SOD symposium, you might remember my advisor, Dr. Ross Mettenmeyer, talking about this idea of tangible, user-friendly models. At the time, this was more of a proof of concept, but since then we've started to work with the stakeholders in Oregon using this kind of participatory modeling framework. So this picture here is our user interface called Tangible Landscape. The idea here is that you can place management on the landscape by placing felt on a representation of the landscape. These felt locations are then used as input for the model, which then runs in the back end, and we display all the disease spread results back onto the landscape. So essentially, this just lets users quickly and intuitively see how that management affected disease spread. Users can switch between different visualizations using the buttons, and all of the data here is stored on a web interface so that you can quickly compare scenarios based on things like cost of management, area infected, etc. So 2 Octobers ago, we held a workshop with experts in the system in Oregon, some of whom are here today. And the goal here was really to get feedback on both this tangible landscape model and the disease model for Oregon to see how we can improve it. The folks who came gave us a lot of really great suggestions, and since then we've been working hard on incorporating them, but I just want to quickly go through a couple of the major things. So first of all, we added yearly management. When we did the workshop a few years ago, you could only manage at the first time step. But in reality, people are going in and managing every single year. So we've changed the modeling workflow so that you can stop the model at any given time and add management. From a GIS standpoint, this is a lot more complicated, but it makes the system more realistic, both for this use and for other uses as well. We also have a collaboration with APHIS, and as part of that, we're developing a website version of this model. The idea here is that it would guide users through all the steps of model development, calibration and validation of the parameters, and then they would be allowed to test out different management scenarios in a similar way to how this tangible landscape works. So right now we're doing this focused on spotted lanternfly spread in Pennsylvania, but we've made a lot of good improvements lately, and we're going to apply the same kind of framework with Southern Devon. Lastly, we've been doing a lot of work on updating the parameters so that they reflect Oregon conditions, both for that NA1 and EU1 strain. Honestly, this was a bit more of a challenge than we were initially expecting. For the sake of time, I'm not going to go too deep into it. But we've made a lot of progress lately, especially for that NA1 strand where we have a lot more data. And I just want to quickly go through some preliminary results of an analysis that we've been working on. So one of the things that I really want to do with this model is use it to generate a hypothetical scenario where Oregon never managed the pathogen, which Sarah mentioned earlier in her presentation. And I think that we could use this to see how effective that management has been in a different kind of way. So first, if you convert all of the point locations of infection into a 300-meter gridded raster type data, you get 26 infected pixels. This is probably an overestimation of the overall infected area, but this is a lot easier to relate to these model results. When we run the disease simulation with the management, we are finding that we're getting about 20 infected pixels out of 100 model runs. So what we're seeing here is that the model is actually fairly conservative. We're predicting less disease spread on the landscape than we see in reality with this management included. However, when you simulate without management, we're getting an average of 110 infected pixels for the same system. So if you convert all of this to area, it seems like the management in that first 5 years of disease spread saved about 800 hectares worth of disease infection. And I think that this goes to show how influential that management really was. Like I said, this was just a preliminary analysis. So our next steps are to expand this for the whole range of time that sudden oak death has been present in Oregon, and to include that EU1 strain as well. And then our other big thing is we have another workshop planned with the Oregon Sudden Oak Task Force, currently at the end of August, where we're going to demonstrate these two tools, the website and the tangible landscape, and all of the updates to them. And I'm really looking forward to seeing how people engage with these tools. Thank you.
Wow, that's right. Okay, hello everyone. My name is Hazel. I'm a second-year PhD working with at Oregon State University, and I'm going to be talking very briefly about site epidemiology and wildlife management in Oregon. So I'm going to assume that you all have remembered everything that everyone has said about the sod in Oregon stuff, and I'm just going to jump right into my specific research theme. So essentially the question that I'm asking is whether or not the sudden oak death management strategies, which Sarah and others have talked about, are effective since the EU1 lineage was introduced. And the reason behind that, obviously, when the epidemic started in Oregon, it was the NA1 lineage, and so EU1 was introduced was first discovered in 2015, but was introduced earlier than 2015. And so We just are sort of following up on the effectiveness of the current treatments in the face of this new lineage that's been introduced. So, just as a brief reminder to people, so this is a continuation of existing research. So we have the inclusion of this EU1 lineage, which you can see here in blue. In addition, we are also still sampling NA1s. We're sampling those that have been treated. We're also sampling NA1s that were discovered but have not been treated in the generally infested area, which was established in 2012. This orange circle here represents the entirety of the quarantine zone. And then up here, this black oval here, that represents uninfested site controls. And so what we've done is, in addition to EU1s that have been treated, EU1s that have not been treated, NA1s that have been treated, and NA1s that have not been treated, we're also sampling tan oak that have no infection whatsoever, that are greater than 8 kilometers away from the nearest infected site. And so that's that black oval up there. In addition, this pink blob here is the outline of the Wildfire, which Sarah talked about, swept through portions of Curry County in 2017. And so in addition to all of this sampling, we're also doing sort of a secondary sampling with the wildfire to see natural impacts of wildfire. Okay, so now I'm going to talk in one slide about my sampling procedure. So it all starts with, in most cases, a red dead tan oak tree. This has been GIS through the aerial surveys and has been tested and confirmed to be positive for some oak death. Next, we set up a nested 1/20th acre and 1/50th acre nested plot. In the small plot, we look for vegetation. We do percent cover and species. And that includes tan oak seedlings. Next, in the large plot, we do DBHs for all tan oak trees. If the site has been treated, then we'll do stump diameters. Next, we do species and DBHs for other standing trees, living trees on the site. Finally, we'll sample 5 of the small vegetations. So specifically, we're looking for vegetation that has symptoms. Often this will be tan oak seedlings. Other than that, we also collect things like salal, evergreen huckleberry, bay, etc. just speaking to the audience. Okay, and then the last thing we do is take 20 soil samples from within the site. And essentially what we do is we bait the soil. So the soil is collected in plastic bags. We moisten the soil to encourage sporulation, flood the soil, and then put a bait bag containing rhododendron leaf discs. After a few days, we take that out, put it on a Phytophthora-specific media, and then subculture. And those subcultures will be sequenced later on. With the vegetation, like you see here in the center image here, the vegetation, we will surface sterilize that and then place that on the Phytophthora-specific media as well to do isolations. So, knowing all of that stuff, I can get to my very specific research question, the first of which are these current treatments effectively reducing ramorum in soil and vegetation for both NA1 and EU1. So the NA1 question has been answered, and the paper was recently published by Hansen on eradication effectiveness for EU1. This is sort of following up on that, and with the addition of the EU1 and all of the other aspects that I talked about earlier. Our hypothesis based on this research and our preliminary results is that treated sites will have fewer remnant ramorum infections in soil and vegetation for both lineages. And then the second research question is whether or not the outcomes of wildfire on Phytophthora ramorum are different than those of current treatments. And the hypothesis is that sites affected by wildfire will have no difference in outcomes. And the reason that I say this is specifically the Wildfire was a variable intensity wildfire, and so a lot of the portions of sod-infected sites that we were able to access were either very low intensity or essentially no intensity. So although the wildfire boundary included some of these sod-positive sites, the wildfire itself doesn't seem to have affected those sites. So far, we've done 70 sites. Hopefully we'll do another 30. This is a very tiny untreated sites. So lots of work left to do. These are some very preliminary data. So I want you to focus on the orangey red boxes here, and that's the vegetation. This is an average of the veg proportion for untreated and treated based on lineage. So you have E1 lineage over here and A1 over here. Essentially what this is showing is that treatment works on vegetation. With the soil we have sort of low sample sizes, and so we're waiting on some more data to flesh out that story a little bit more. And with that, funding provided by Forest Service, ODF, Oregon State University. I'd like to thank all these lovely people here, some of which are in the audience, and I think that's it for me. Thank you.
Okay. Oh, shoot, mine's not in here. Sorry, I did mine in 4 to 3 instead of 16 to 9, and so I was afraid of like everything moving. So just take a little moment to load that. Okay, there it is. And everything's in the right place so far, so that's great. So I'm going to start off by just acknowledging everyone who's helped me get to this point, especially, well, everyone listed. I've probably forgotten a bunch of people, and I didn't list every single person who's ever worked on our plot network. There's a lot of them, and the data I have wouldn't be there if not for them. So I'm Jacqueline Rose, forgot to introduce myself. I'm doing a master's program at the University of Cal Poly in San Luis Obispo, California. And my focus is on the vegetative community as a whole in Big Sur and how it's changing due to Phytophthora, ramorum, and wildfire. And so I realized that I was kind of losing the forest for the trees and looking at all these big, beautiful like redwoods and tan oaks and not looking at what was at my feet and wondering like what's happening with the understory and what's happening with the shrubs and the herbaceous thing and the grasses. So I have 3 different hypotheses that I'm going to talk about today that I did some preliminary work on and have some results to share. So my first hypothesis is that the abundance of nitrogen-fixing genera, which are Fabaceae genera and CNPS, will be increased immediately following fire. You'll notice that doesn't mention Phytophthora ramorum. I'm hoping to connect those later in my research, but they aren't connected right now, unfortunately. My second hypothesis is that the incidence of non-native species is going to be greater in plots that are positive for Phytophthora ramorum prior to a fire. And my third one is that diversity is going to be lower in plots that are positive for Phytophthora ramorum. So the network that I'm using is one that was set up in 2006 and 2007. Carrie mentioned it earlier in her talk, and we have 280 plots in Big Sur, California. It's located in Monterey County that are all along the coast. They cover a variety of different categories, I guess. So we have mixed evergreen plots, which you can see in the photo on the right. We have redwood plots, which you can see in the photo on the left. And those are represented by blue and red dots, respectively. And we have plots that are infected, which are the closed circles, and uninfected, which are open circles. You can see those. They're also on like public and private land and at a variety of elevations. And there's a bunch of different things that are going on there that I actually didn't use in this research so far, but we'll hopefully get into later. And then this was originally set up to look at just Phytophthora ramorum and its effect in Big Sur, but then about a third of the plots burned in 2008, and then a third of the plots burned in 2016. And so now we have this really interesting experiment where we have data that was prior to the 2008 fire, between the fires, and then in some plots that have been burned twice, like only 8 years apart. And so the data that I'm specifically looking at is cover class data, where we looked at every single species we found on each plot and gave it a cover class between 1 and 7, based on just how much area of the plot that it was covering. And so we have over 650 plot surveys and over 13,000 records of individual plants on those surveys. And only one listed species showed up, and so that's Aedes bracteata, the bristlecone fir, and that's listed by CNPS. It's not listed federally or by the state. And so for my first hypothesis, that the abundance of the nitrogen-fixing genera will be increased immediately following a fire, it's not statistically significant, the differences here. So I have along the bottom row years since fire, and I've kind of put plots into different bins according to when we surveyed after the fire. And so in the 0 to 2 range year, you'll see that it's pretty low. There's almost no coverage. That makes sense. Everything's recovering from the fire because everything burned. And then from the 3 to 5 year, it's still low for both the mixed evergreen and the redwood, but you'll see that the max is the highest of any of them. And so there's a lot of variation going on. And it might be that there are some things in our plot network that I'm not controlling for that will explain like that variation and that this might be significant. But right now it appears that it's not. And so my second hypothesis that non-native species, there's going to be more of them in the plots that are affected by Phytophthora worm prior to a fire. You'll see that the uninfected ones, which are the black bars, over 50% of them didn't have a non-native species that we recorded in the plot. And so that decreases as you increase the number of species. So it's more likely on uninfected plots that you won't have any non-native species. And then for the final one, that diversity will be lower in plots that are positive for Phytophthora worm. I did not find a significant difference here between anything, so between like mixed evergreen or redwood, or between the Phytophthora worm positive and negative plots. And so again, it's possible that there's something that I'm not controlling for that could, show that there is a difference, but right now it looks like there isn't a difference in diversity. So in conclusion, my hypothesis that nitrogen fixers will increase following a fire is possible. That there are more non-native species in infected plots, also possible. And that diversity is lower in infected plots, looks like that's false. Thank you.
Today I'm going to be talking a little bit about the impacts of wildfire and wildfire history on the epidemiology of Sonoma. Oh, I'm missing a slide. Okay, well, I'm going to go from here. So the fire, all of the forests that are invaded by Phytophthora ramorum are fire adapted. They're prone to frequent fire, and the species present in these forests have important adaptations to wildfire. For example, in these forests that you see on the left, following a wildfire, most of the dominant tree species we have in the Big Sur region can resprout. And so when the overstory canopy is removed, the above ground stems are damaged, you see this prolific post-fire regeneration where these species regrow asexually really quickly. And considering fire's important changes to canopy structure, it stands to reason that wildfire might actually play an important role in determining the patterns of sudden oak death that we see on the landscape. But the very few studies have examined the mechanisms actually determining that trend, and if that trend is robust across a wide gradient of fire history. In my missing slide, I was going to point out that following wildfire in the Big Sur region, we found that wildfire doesn't actually remove the pathogen. It removes the pathogen in places where the overstory canopy has been consumed, but in places where the canopy remains or individual trees have persisted, the pathogen can survive. And so we wanted to look at essentially how, I'm missing quite a few slides, so so essentially our questions were to better assess the extent to which wildfire and wildfire history influence different dimensions of the epidemiology of sod, including not only the occurrence of the pathogen, but also how it spreads or intensifies within stands, and also its effects on patterns of host mortality. So essentially how sudden oak death acts as a disturbance on these landscapes. And to examine that, we're using that same long-term plot network that Jacqueline just described, where there were 280 forest plots present across the Big Sur region. And in addition to representing a gradient in disease occurrence and disease impacts on the landscape, these plots also represent a gradient of past fire history since the 1950s, as captured by the California Fire Perimeter Database and shown on the different colors of the points here. This plot network has also captured the effects of some recent wildfires, including the 2008 Basin Complex fire. And so we wanted to take both of these sources of data, the effects of long-term fire history and the effects of this recent wildfire, to explore whether there was a relationship between fire history and the subsequent occurrence of sudden oak death and how it functions as a disturbance in the landscape. Specifically examining hypotheses that relate to how it might influence the spatial relationships between hosts, the composition of hosts, and microclimatic conditions within the stands. And so when examining that long-term fire history data, we assessed the extent to which that fire history influenced sudden oak death dynamics that we saw in the Big Sur region. Most of the models that I'm going to describe today include variables for climate, but I won't talk about that further. And we included information about the two different forest types that Jacqueline just described, that mixed evergreen and that redwood forest type. And what we found was that with increasing fire frequency since 1950, we found that Phytophthora ramorum was much less likely to be present at the stand level. We also found that in places where Phytophthora ramorum had colonized, that the stand level infestation intensity decreased with fire frequency as well. And we found that increasing fire frequency decreased the amount of host mortality as measured by basal area on the y-axis there, suggesting that wildfire plays some role in determining the severity of sudden oak death as a disturbance on the landscape. So generally, frequent fire seems to dampen disease dynamics. So next we explored which mechanisms actually determine that trend. And what we found was that generally, in the same areas that had burned more regularly, we found this decrease in overall host basal area in terms of those primary sporulating hosts, the tan oak and bay. We found a general decrease in bay laurel basal area. And we found that tan oak's likelihood of being present at the stand level also decreased. And this makes sense because among the species present in these forests, these are some of the species that have the thinnest bark and are most likely to be removed by wildfire. And so then we used the recent wildfire in 2008 to explore whether the impacts of a recent wildfire corroborated any of these mechanisms, specifically examining which factors, which aspects of stand composition and stand structure were influencing the spread of Phytophthora ramorum into these burned areas. As I mentioned previously, the pathogen primarily persisted in those intact canopies, host canopies that had been left behind by wildfire, like you can see here. But there's also a ton of resprouting regeneration that occurred really rapidly following this fire. This is only 5 years out, and most of these shrubs are over people's heads. And what we found is that in these burned areas, the primary driver of reinfection of hosts, or spread of the pathogen back into these burned areas, was whether there were surviving bay laurel stems, and larger stems tended to more positively predict the rates of host reinfection. Interestingly, even though there's all this post-fire regeneration from bay, from tan oak, we found that the resprouting host basal area played almost no role, suggesting that there might actually be something really important about the vertical structure or the microclimates provided by these surviving bay canopies. We hypothesized that the vertical structure of these hosts actually allows the inoculum to rain down and disperse horizontally in a way that these, even though they're very well connected, relatively short resprouting hosts do not. And so we think that these impacts of host structure, rather than strictly host composition, are determining this relationship between wildfire and sudden oak death. And here are my other slides. This is just some pictures of the resprouting regeneration. Oh. All right. So the essential question is, if sudden oak death or Phytophthora ramorum occurrence and possible spread is decreased in these recently burned areas, does it make a difference? Does it matter to these host tree species? And so we constructed a model of host stem mortality, comparing patterns of host stem mortality in tan oaks and oaks to areas that had been burned and versus unburned in this recent 2008 wildfire. And what we found was, this is showing the parameter estimates from that model. And what I want to draw your attention to is that the effect of Phytophthora ramorum infection positively increased a stem's likelihood of dying, just what we would expect in these unburned areas. But in the burned areas, that effect of Phytophthora ramorum infection actually seems to go away, suggesting that that the effect of Phytophthora ramorum infection is diminished in these burned areas. And we think that this has to do with that same decrease in kind of the inoculum pressure in these recently burned areas, suggesting that these hosts might actually be temporarily released in burned areas. And so I think these have important implications for sudden oak death management. We found that frequent fire dampens disease dynamics and might actually play a role in predicting where disease acts as a disturbance and how severely it acts on the landscape. That this trend is mostly determined by base structure, and that these host trees seem to be temporarily released in these recently burned areas from pathogen pressure. I think that this has important connections to the conversation around whether prescribed fire might play a role in sudden oak death management. I think that the important driver here suggests that removing base structure using prescribed fire would only be effective in terms of its ability to remove base structure and take those bay trees down to ground level. We weren't able to assess the importance of microclimatic effects in this analysis because you can't separate those from the host canopies, but we didn't find general effects of canopy cover overall, suggesting that the host composition and the host structure is more important than microclimatic effects overall. And I think that this is important to consider, considering that many of these landscapes that we've talked about today are predicted to be more frequently burned or more severely burned in the future. And so I just want to say thank you to everyone who helped collect this data, and I don't know what happened with us.
Okay, so for the Q&A, if I could get all the people who just presented to come on up. And then when you guys ask your questions, you can direct it to whomever you want, or we can have a scrum.